Okay, thank you everybody for joining the August 2025 Alliance office hours and webinar. Today we have Chris Mungall, who's one of the PIs in the Alliance, who's going to talk to us about ontologies and how they're used at the Alliance. Just a reminder that in September, so next month, September 25th, we're going to have a guest speaker, a non-Alliance member, Damien Smedley, who's professor of computational genomics at Queen Mary University of London, is going to give a talk on the use of model organism data in rare disease diagnostics and disease gene association discovery. So that should be really good, and I hope you'll all register and come back for that presentation. So Chris, thank you again for giving a presentation today, and I'll turn the floor over to you.
Okay, thank you, Carol. Yeah, so I'm part of the Alliance. I'm one of the PIs on the Gene Ontology project, and the Gene Ontology is, of course, a part of the Alliance. So I'm going to be talking about how ontologies are used in the Alliance. So, I'm going to keep this very much a practical walkthrough about specifically what ontologies we use in the Alliance, why we use them, and then a little bit more on how you can get additional information on these individual ontologies, how you might be able to use them yourselves to power more advanced analyses, and just a kind of a bit of an outlook on how ontologies relate to AI moving forward. But yeah, just to kind of I think looking at the crowd here, I think everyone is fairly familiar with ontologies. But here's my kind of brief 2-slide intro into what really they are and why they use them. So you can find many different definitions of ontologies on the web, but for our purposes in biomedicine, mostly ontologies refer essentially to a network of linked terms or descriptors, each of which describe some concept in an area of biology. And there are different ontologies covering different parts of biology. This could be like gene functions or cell types, anatomical structure, disease and phenotypes. And there are in fact many more. And we'll cover some of the ones that are used in the Alliance. And ontologies can be used for really quite a broad range of different purposes. But we're going to focus today on one of the main use cases of ontologies, and that's really for being able to annotate or tag different kinds of named entities, named biological entities. So this could be genes or alleles, could even be something like an experimental sample where you want to annotate it with its tissue type. And I won't go into too much detail about how these annotations are made, but it's just useful to know that they can be made via a variety of different methods. Of course, at the core of this, there are manually curated annotations made by biocurators who will read the literature and read some kind of like a functional description of a gene, or maybe a description of where a gene is expressed, and they will manually annotate that. Annotations can also be propagated using bioinformatics means, particularly for something like the gene ontology. You might have something like a hidden Markov model or some kind of phylogenetic model of a protein and its structure, and from that you're able to, in some cases, reliably infer what its function is. But increasingly, people are looking at using AI for that to assist with that manual annotation. And I like to show things with these graphy type images here, so you can see a collection of these terms, like this term from the Go, cysteine type endopeptidase activity. It's arranged in a network. It's got 2 parents, and then we've got a gene or gene product colored in blue on the left, and that is essentially tagged or annotated with the term. So I'll go into just a little bit more detail about what an ontology term is and what you might expect to find or not find there. So this is my anatomy of an anatomy ontology term. So the idea here is that we are trying to build some kind of a model or a set of descriptors for maybe a part of an organ system like a kidney or a glomerular capsule. I'm going to call that a Bowman's capsule because that's a synonym, as you can see in this little box here. So one crucial thing to know about all of the ontologies we use in the Alliance and more broadly within the biomedical community is that they follow FAIR principles, and we'll get to the Obo Foundry principles in a moment, but every single concept has a unique identifier. We call this a query. So it's a two-part identifier. It's got the namespace and then some kind of semantics-free identifier. That can also be translated into a permanent URL or PURL. And crucially, for most of the ontologies we use, these come with definitions, because it can be misleading just to try and understand what a term represents just by using the label, because we often use the same label in different ways or different labels in the same ways. And then because there's often different systems for representing the same concept, we often have kind of like mappings or kind of cross-refs, as we sometimes call them. So you can see this in the box on the upper right here. And then these terms are interlinked together in a graph using edges, and the labels on these edges also come from ontology terms as well. So we make use of the relation ontology to be able to express things like Bowman's space is part of Bowman's capsule, or that Bowman's capsule develops from something like the L-shaped body. So these terms are from the Uberon ontology, which we'll get to in a moment, but just to kind of really anchor it in the Alliance website and how you can actually, make use of these yourselves. One thing that should be fairly obvious to people coming to the Alliance site is that ontologies are really used quite ubiquitously in the Alliance, and this is obviously true for many biomedical knowledge bases. You would see the same thing in to some extent in Uniprot, but I think the Alliance really makes quite heavy use of ontologies to structure the information. And so one way to see that is if you're on a page for a gene like caspase-1 in mouse, the Alliance has this nice little index on the left-hand side where you can zoom in and find out some information about some particular aspect of that gene. And each of these aspects, not all of them, but many of them are essentially guided by an ontology. And we'll kind of go through some of these. So function using Go, for example, fairly obviously, but maybe other aspects as well, like where the gene is expressed, might make use of a different ontology from Go. And you may not be aware, but there's in things like the Alliance automated gene descriptions as well. The ontology annotations are actually used here, and they're used algorithmically to generate a nice natural language description of different aspects of that gene. And so you can see here I've highlighted a sentence: enables cysteine type endopeptidase activity involved in several processes. So this is a nice natural language statement, but this term is actually in fact from the Go. And then further on in the description, you can see maybe what are terms from other ontologies, like the small intestine, for example. So these ontology annotations all go into making these natural language gene descriptions. So I'll walk through some of the different components of the Alliance view and how these are used, how ontologies are used in each of these. So if we were to, now of course not every single one of the aspects in the Alliance makes heavy use of an ontology. There's obviously very useful sections in the Alliance site for looking at orthology and paralogy, but if we were to scroll on down to function and Go annotations, you can see what we don't do here is just overload the user with a giant list of all of the different Go terms that have been used to annotate the gene. That can sometimes be a little bit overwhelming. So we make use of this widget that was developed for the Alliance called the ribbon. And this is the Go ribbon, but the ribbon technology is used it's generic. It can be used for other kinds of ontology annotations as well. And what this does is it makes use of a concept called an ontology subset, or in Go, we sometimes call these slims or Go slims. And the basic idea is that a curator can define ahead of time a set of high-level terms, maybe things like catalytic activity or enzyme regulator activity. And then what we can do is use this to roll up more specific annotations to these more general terms. And you can do nice things like highlight the density or the number of terms that have been annotated in each of those rows. And then what you can do is you can then zoom in on some of these areas if you want to know more details. So what I'm showing here is you can see in the ribbon view, we're still on the CASP1 gene here. The user has clicked on the box in the ribbon for catalytic activity, and they can see the more specific term here, cysteine type endopeptidase activity from the Go, along with all of the evidence that goes to make that association. And it's kind of ontologies all the way down. I don't have any slides on this, but you should know also that these evidence types as well come from an ontology called ECO, the Evidence and Conclusion Ontology. So not only do we use it to tag the term itself, we use it for having complete provenance about how the annotation got into our database. So in the case of Go, you can always explore more about that individual Go term. You can find a list of all genes in all species that might be annotated to that, or if you just want to get more information about the term itself, you can click on that term and it will take you over to Amigo, which is a browser used for within the Go consortium for looking at Go terms, and it also has links out to the EBI Quick Go tool as well. So you can see here the anatomy of the Go term here, cysteine type endopeptidase activity. You can see it's got a number of different synonyms. You can see it's got a full text definition. And then on the Amigo page, you can also look at genes in other species as well. So if we were to kind of scroll down further in the page, this is one of my favorite parts of the Alliance view, is you can get these wonderful views of how that gene might be active in different pathways. And we have a few different tabs here. We've got a couple of reactome tabs, which I don't have time to go into, but I think you should definitely go and check those out. But what I'm showing you here is a tab with some slightly newer pathway annotations that come from the Go consortium. And we call these GoCams, or Go Causal Activity Models. And the basic idea here is what we've been doing is we have been taking existing Go annotations and essentially linking them together into a broader causal model of what that gene is doing in the context of what other genes do. So with a standard Go annotation, you might be able to say something like CASP1 is a cysteine type endopeptidase activity, it's in the cytosol, and so on. But with a GoCam, you can link this to its downstream target, which in this case, might be another gene, and that then activates CASP7, and so on and so on. And for these more complex pathway models, we are making use of ontologies, not just the Go, but other ontologies as well. So I've highlighted the CASP1 gene in the right here. You can see all of its standard Go annotations. Now, crucially, these are all grouped together. So these make essentially a compound statement. So we're saying that CASP1 has cysteine type endopeptidase activity in the cytosol of an intestinal epithelial cell during this protein maturation process. And what you'll notice here is that we're making use of another ontology here as well. The boundaries of Go at the anatomical level lie essentially at the cell. When we want to describe particular types of cell, we make use of the obo cell ontology here. And this is a hierarchy of types like intestinal epithelial cell and so on. And one thing that GoCams also allow you to do is to describe what is happening metabolically as well. What are the products of different kinds of reactions catalyzed by the different genes? And we make use of the Kebi ontology for that. And you can see this in the bottom part of the GoCam diagram here. So moving on from Go, which of course describes the normal evolved wild type function of a gene. we get into another part of the Alliance where we make use of phenotype ontologies, and this is for being able to describe what happens if that gene is perturbed in some way. And in the Alliance, we have phenotype annotations at the gene level, but also at the level of individual genotypic components like alleles and so on. But just for simplicity, I'm showing you things just on the gene page at the moment. And one difference here with Go is that for being able to describe phenotype, it's useful to get into the specifics of the individual organism that you are capturing. So what we've seen is that there are a number of different ontologies that are used for different species or for different taxa within the Alliance. So I'm showing you here phenotypes for Cas1 in mouse, which is making use of MP, or the mammalian phenotype ontology developed by the MGI group. If we were to hop over and click on the ortholog view and jump on over to the ortholog in zebrafish, CASP-A, or one of the orthologs, we'll see that they also have, we also have phenotype descriptions for what happens if you mutate that gene in zebrafish. But these are using ZP, or the zebrafish ontology. And you can see here there's a large range of different phenotypes that are caused by mutating this gene. Now, one thing you'll notice is at the moment in the Alliance view, we're just showing these as if they are text terms. But you should know that under the hood, ontology term IDs are being used to model these, and these will be visible in future versions of the Alliance site. So is indeed kind of phenotype ontology terms as well. Then another aspect of what happens when genes are perturbed or knocked out or mutated or varied in some way is what kinds of diseases might arise, both in what kind of human diseases arise with mutations in the human gene, but also how model organisms and model organism genes or alleles might be used to study those diseases. So on all the gene pages in the Alliance, whether it's a human gene or whether it's a model organism gene, you can find a list of disease annotations where we have curations of in human what happens if what studies have been done on kind of like variants of that gene and how they might relate to human disease, or indeed models like in this case the rat Cas1 gene. And for disease annotations, we make use of the DO, or the human disease ontology. And again, these are fully modeled with the IDs. You can kind of click on any one of these. And for diseases, we have actual term pages within the Alliance. So and of course these will link out to other representations, such as the OBO site or the DO site for this. But what I'm showing here is how we how not just in Go, but for other ontologies as well, we can make use of this concept of rolling up to a broader concept. So this is the DO page for synucleinopathy, which is a neurodegenerative disease that encompasses things like Parkinson's disease, as well as a range of other diseases as well. And you can see here that even though the annotations of these genes, like A2M and ABL1, is to the more specific Parkinson's disease, they all get rolled up, and you can see them in the context of all synucleinopathies on the Alliance term page here. And yes, sorry, yeah, this is just illustrating the kind of like the roll-up concept. The term page will actually tell you what the direct children are, and you can see that the actual genes are annotated to those direct children. So moving on to the expression part of the display. So in this case, what we do is we make use of anatomy ontologies. And so again, we're back on the mouse caspase-1 gene here as well. You can see this particular gene has been annotated via RT-PCR to expression in the small intestine. And again, because there's a lot of variability across different model organisms, we've elected to use, in many cases, but not all, species-specific anatomy ontologies. So in the case of expression, the EMAP-A, I'm not going to spell that out, the naming is there's some historic reasons for that, but yes, that's the mouse anatomy ontology that contains the term small intestine. We'll come to later how this can be integrated with other ontologies, but crucially, I did not mean to. Okay, that's a mistake in what I. Obviously, a mistake in what I highlighted here, but I intended to highlight the column to the left of that, which is the stage column, Tyler stage 26, which itself comes from a different branch of that stage ontology. So these anatomy ontologies tend to either come in twos or they have two branches, one describing the structures themselves and the others describing the actual developmental stages, not the assay, as I've incorrectly highlighted there. And again, if we were to hop over onto zebrafish, again, we can see, same mistake here, but basic idea is that there's actually two ontologies here technically, but they can also be merged into one. And the crucial part is that there's two branches, one describing the structures. In this case, it's expressed in the intestinal bulb in zebrafish, and it's at larval day 4. And so similar kind of like idea, and again, making use of a more species specific anatomy ontology for describing structures that might be more specific to individual organisms. So ontologies also power search within the Alliance as well. So if you're interested in a general kind of biological phenomena, or you want to zoom in on maybe some kind of like anatomical structure, but you're not sure exactly what aspect it is you want to zoom in on, you can come to the Alliance search page and type in something like cerebellum, hit return, and you'll immediately see a variety of kind of like genes for which that might be associated, but also the relevant ontology terms. And in this case, you'll see cerebellum structural organization, which is a process within the Go, cerebellum cancer, which is a term within the DO. In the future, you might actually see the Uberon term or terms for the actual structures themselves. But at the moment we're just only exposing a few of these ontology terms in the search. And you can see this general concept of auto roll up here as well. If you were to dive in and get all of the genes annotated with this term, it would also roll these up and get genes that are annotated to a more specific is it or part of child as well. So how do these pieces all fit together? Well, for some of the species-specific ontologies, such as the mammalian phenotype ontology, the human phenotype ontology, or the zebrafish phenotype ontology, there is a kind of bridging ontology called UPheno that was developed as a collaboration between the Alliance and the Monarch Initiative. And this essentially gives you a set of species-neutral phenotype terms that can that essentially act as a children to the species-specific, as a parents to the species-specific as a children. Then for expression, there is the Uberon ontology, which has been around for some time, which was developed originally as a way of being able to bridge different anatomy ontologies, but has now evolved into more of a fully fledged cross-species ontology, at least for metazoa in its own right. And of course, ontologies like the gene ontology are composed of different ontologies, the molecular function, biological process, cell component ontology. And these ontologies are all actually interlinked together as part of a knowledge graph. So this is an example from the cell ontology. This is color-coded, showing different ontologies. So we've got the cell ontology in yellow, and you've got links going out. The cell ontology has part links, going from cell types to particular subcellular structures that might be found in the Go cell component ontology, like microvillus. We're also effectively annotating cell types in the cell ontology using Go terms as well, although they're not actually captured as separate annotations. They're captured directly as edges within the ontology. So you can navigate from a term in the cell ontology like hippocampal astrocyte to things like ion homeostasis that describe either its function or some of the processes that that cell type is involved in. And of course, you have links going from the cell types up to the broader anatomical structures which go in the partner ontology Uberon. So traversing from hippocampal astrocyte is a part of the hippocampus, for example. So all the ontologies used in the Alliance are part of OBO, or the Open Biomedical Ontologies Foundry. And so the basic idea with OBO is it arose, many years ago, originally as an offshoot of the gene ontology, when in the gene ontology we realized that we needed to make use of other ontologies to describe other aspects of biology. A lot of the original ontologies came from Flybase. Michael Ashburner was like instrumental in developing a lot of these, and the OBO has since grown into both. It's a social mechanism and a technical mechanism as well. We use this to develop standards to help all of these different ontologies work together, and we also built tooling as well to help people build the ontologies and make them conform to the standards. So, this includes tools like the Ontology Development Kit, which is now, the de facto standard way of both getting started with building your ontology and also integrating it into a broader ecosystem and bringing in QA and QC mechanisms that can be automatically integrated with GitHub and so on. And one of the key parts of OBO is this notion of the OBO principles that allow us, to actually kind of like concretize this concept of fair, findable, accessible, interoperable, and reusable. We have our own set of principles essentially for OBO ontologies, and we have kind of dashboards that show, which ontologies conform and so on. And you can explore this on the OBO site itself. So if there are ontologies that are being used in the Alliance, you should of course use the Alliance APIs to download those annotations. But if you want to dive into more detail in any of the ontologies themselves, you can go over to obofoundry.org and browse some of these ontologies in the repo. This also gives you a standard way of being able to find things like what's the GitHub issue tracker for this ontology, because I want to go and request a new term or request a replacement of an existing term, for example. So I really encourage people to use OBO as a standard way of doing that, rather than just trying to randomly Google for some of these things. And so OBO works in concert with existing dedicated ontology browsers, such as the OLS and BioPortal, which, unlike the Alliance term pages, don't actually show you the annotations to these genes, but this can give you a deeper, kind of like a more granular view as to the actual structure of some of these ontologies themselves. And each of these sites have their own kind of like unique features and ways of being able to navigate and explore these ontologies. So, a key recent part of OBO has been this offshoot with some funding from the Critical Path Institute and Monarch, with many contributors from the Alliance. And what we've been doing is realizing that not everyone is necessarily as facile as members of the Alliance are when it comes to working with and using ontologies or developing ontologies. We recognize the need for better training material. A lot of the training material for ontologies out there on the web is written by maybe kind of like computer scientists or formal ontologists. We wanted a more practical, easy to use guide on, just simple things like how do I go about requesting changes for an ontology term. So it's really intended to be, a sort of like a day-to-day practical guide. It is kind of constantly in progress. So if you see things here that you think there's something missing, then just make a link in our GitHub issue tracker. And we've got many examples of how how to use things like some of the Alliance ontologies in here. So going back to the Alliance, I think there's been a previous one of these office hours on the APIs, but just to really emphasize for computational users, the APIs really, ontologies really underpin a lot of the payloads that you get back from the APIs. So I'm showing here one of the API endpoints that you can use to computationally retrieve all of the expression information for a particular mouse gene. Again, this is back to Caspase-1, and you can see in the payload that gets returned for the Ruben part of this, it's got the Uberon term for alimentary part of the gastrointestinal system. And this really helps make our APIs fair, in contrast to if we just returned strings for each one of these. The data that comes back from these APIs would not be interoperable with other databases that come from other parts of the NIH ecosystem. And you can see this as well in the ontology downloads, enabled downloads that we have. So for pretty much everything that you see on the Alliance site, you can come along and download either JSON files or tabular files that you can look at in Excel. And again, we always make a point of including the ontology identifier along with the label for human readability, of course, in some of these downloads. So you can see here, these are all of this is all of the a subset of all of the disease annotations to DO terms in the Alliance. And you can see where we're associating not just genes, but alleles and genomic models with some of those terms as well. So just some additional tooling that you can make use if you're a computational user. Of course, there's many different programming languages that people use these days, but I think a lot of people are making use of Python. We have this tool called OAK, or the Ontology Access Kit. It's a companion to the Ontology Development Kit. There's a Python library for being able to query and make use of ontologies and use it for analyzing your data. And you can use it either as a Python library, or even if you're not a Python programmer and you're comfortable using the terminal or the command line, it includes a lot of quite powerful commands for being able to query ontologies on the command line. And you can use this in concert with things like the downloads or the payloads that you get from the Alliance API. So what we're showing here in the middle is how OAK can be used on the command line for doing some powerful operations over, in this case, the Drosophila anatomy ontology, which is the namespace FBBT. And this is just showing how you can use commands to draw a tree, for example, going from a particular term like the wing vein all the way up to the root of the ontology. So I encourage you to check that out and explore more, whether you're a Python developer or you just want to use some powerful ontology tools on the command line. So I'll touch briefly on this, but this is an area for future development in the Alliance, as well as being able to simply do lookups using ontology terms. One of the key use cases for ontologies is to essentially power high throughput data analyses. So a classic example here is an enrichment analysis where you might have a set of genes that are overexpressed or underexpressed in a particular condition, and you want to know what characterizes those genes and what makes them different from the background set. So one of the most common use cases here is to do a Go enrichment analysis. Right now, you can use the Go site. Go is of course part of the Alliance, and you can use the Go site, powered by the Panther database, to do an enrichment analysis on either your human or your model organism genes. We're also working with other groups to allow you to do these enrichment analyses using pathways as well. So we have a partnership with the Index group, where you can go there right now and you can do enrichment analyses and see your genes lit up on pathway diagrams. We have that working for humans, but not model organisms. Now, what if you want to do an enrichment analysis, but you want to see not just Go, but you want to see maybe what phenotypes or diseases are enriched? Right now, that a lot of that functionality is still mod specific, but we're looking into ways of bringing this into the Alliance, making use of some of the Go tools here as well. And of course, enrichment analysis isn't the only analysis you can do. Many people make use of a concept called semantic similarity analysis, particularly for doing phenotypic use cases, like trying to match a set of phenotypes for a mouse gene to a human disease. And you can make use of external sites like the MARC initiative that make use of the Alliance Ontologies and bridging ontologies like UPheno and the Alliance annotations to drive that. And you can hear a little bit about that in the session that's coming up in a month. But anyway, so yeah, at the moment, you can't do all of these directly within the Alliance site, but this is something that we have planned for the future. And just to kind of show you a little bit about how the sausage gets made, this is a screenshot of the new curation tools that are being used in the Alliance. And again, all of these are heavily ontology driven. We make use of a set of common generic widgets for being able to allowing the curator to easily navigate through ontology terms to make, for example, a disease annotation. They can just type in a text box, type in the name into a text box and get back all matching terms and do a little bit of kind of exploration of the ontology directly there in the curation tool. So this kind of shows really just how this is kind of underpinning all of the annotations that we have. So just moving on briefly to a quick kind of perspective on AI and how within the Alliance, we have an Alliance AI working group that is looking at ways we can use AI to turbocharge some of our general annotation processes, being able to curate gene annotations from the literature. But we're also exploring ways in which AI can help us actually with the actual ontology building themselves, because these ontologies, they take a lot of resources to build and maintain over time. We constantly need to go back and revise them. It's not just adding new terms, but we need to move terms around. And with newer kinds of agentic AI technology, it's actually becoming possible to do some quite complex AI workflows, even directly from just from GitHub issues. So this is a hypothetical example here where a user or a curator might want to add multiple terms to the cell ontology corresponding to all transcriptomically identified molecular layer interneuron subtypes. And here, using agentic AI, the AI can essentially carry out a number of different subtasks and chain them together. And this might involve things like looking at the existing structure of the ontology, making use of tools like OLS, downloading publications, reading those publications, making edits to the ontology, running deterministic QC checks over that, running reasoning, and then essentially making a pull request to that ontology. And we're actually deploying this on a number of ontologies at the moment. This is an example from the Uberon issue tracker, where a curator, actually a curator who used to work on Go, asked for a new term, sp-sclerovein. Now, in this case, the curator did the huge majority of the research work up front. They researched a good publication, or at least in this case, a Wikipedia reference to use for this, and they figured out where it should go in the ontology. But it's still a bit of work, and in this case, kind of quite tedious work for an ontology editor to open up the ontology editing tool and make the change and so on. So we want to explore ways of being able to automate kind of little tasks like this. In this case, the Copilot agent was able to essentially read this issue and go off and make edits to the ontology and make the pull request. So in this case, all the curator has to do is check the changes that the AI provided. And we're in the process of evaluating this. There's definitely some things that works less well on it, but for some things it works really well, and we think this can be a really key way to helping us automate some of these more mechanical tasks in ontology building. But in addition to AI being useful for us, ontologies, I think, are clearly vital for AI moving forward, because it might seem that we have these new big genome foundation models now. They're able to just process, they are trained on huge amounts of often unlabeled genomic data, but the real value in them comes when you fine tune these models, making use of curated annotations of the kind that we've seen in the Alliance. So this is a screenshot here from a recent paper on alpha genome. What the alpha genome model allows you to do is to essentially predict the regulatory effects of mutations. But I think what is key and really cool about this particular model that other models before it weren't really able to do is it's able to do this in a tissue-specific or cell type-specific manner. And in order for this to work, they made heavy use of ontology labeled annotations from like RNA-seq data and so on, where curators had painstakingly made sure that all of the different samples that came in were tagged using these terms, such that they're comparable, such that they can be used to train these models, but also they can be used at inference time as well. So what's really cool about this model is they have some example Jupyter notebooks, and you can actually go in and you can actually say, give it a CL ID or an Uberon ID and say, and a particular mutation, and say, what are the effects of this mutation in this tissue type? So this is all made possible using ontology annotations. And I think just with that kind of like future looking thought, I'll leave it here and can take any questions. And again, just to highlight the next talk coming up, which touches on some of the themes I covered here, which is from Damien Smedley on using model organism data, and in particular phenotypic data for disease gene association discovery. And with that, I will take any questions.
Thank you, Chris. There is a question in the chat, actually two questions. Question number one, is the Alliance connected with the Human Phenotype Ontology database?
So I think someone on the Alliance can just, I believe it's similar to MP and so on at the moment where we are, but I don't think we're pulling in the
Chris,
HPO
Actually, we do.
we are.
We do, yes.
Yeah.
Sorry.
Via RGD.
Yeah.
Yes, we have annotations for human genes to the HPO ontology. Most of them come from the HPO group, and some of them have been manually curated by RGD.
That's right. Yes. And also, I meant, this will be covered a little bit in Damien's talk as well, I think, but, there are different phenotype ontologies used for different species, but the eupheno bridging ontology can be used to traverse across those. So that you can say something like, what are all the lung phenotypes across all organisms, and it can make use of, it will automatically roll up HPO lung annotations and MP lung annotations and Xenopus lung annotations from the Xenopus phenotype ontology and so on.
Great. And the second question in the chat, based on Go, can gene expression perturbation, say in an in vitro study, be associated with cell phenotype? Say, for example, potential perturbation is associated with a negative effect. Hopefully I've read that correctly.
Yeah, maybe I'll see if someone in the one of the Alliance curators. I think that's maybe a question about how we might curate kind of like expression phenotypes, if I understand correctly. Does someone want to? But yeah, I mean, maybe I'll kind of just give my perspective as well. That I think even though we may curate some of these things independently, often they work together to kind of give us a bigger picture of the function of the gene. So it's often by looking at all of the different aspects of the gene on the Alliance page and seeing where it's expressed and maybe, conditions under which it's expressed along with the function and the phenotype. So you start to kind of build up a broader picture of what that gene does.
So the clarification is asking about gene to phenotype association through the Go.
Mm-hmm.
Yeah.
So we have, of course, in Go, we have the IMP phenotype. We have, Go associations that are made from mutant phenotypes. So you will see some of the Go associations of this IMP evidence code. And, it might be something like if the mutation, leads to a defect in cerebellar development, then you might have a Go term for cerebellar development. And you also have kind of like inferences that are made more at the cellular level as well. Now, of course, you can, what I didn't go into today is I showed you an example of the cell ontology and how the cell ontology links out to other ontologies. This is also true of the phenotype ontologies. So for that particular example, like maybe in the mammalian phenotype ontology, you'll find a term like abnormal cerebellum development. And in the mammalian phenotype ontology, it's a nice kind of tree-like hierarchy, but there's sideways links pointing out. In that case, it might point out to a Go term like cerebellum development. So we have the two aspects of the biology, the normal evolved function and the mutant phenotype, and these are actually kind of linked through these cross ontology axiomatic links.
Chris, I actually had a question as well, which is the GoCam models that you mentioned. So is there a way to look at similarities and differences of those models across different species, like in an integrated way, or do you have to visit each GoCam model that might have been built for a different organism and compare them manually? The reason I'm asking is there are some pathways that have pieces that are different in humans and model organisms. And I'm wondering if there's a way to do that sort of comparative framework of the GoCam models.
Yeah, no, that's a really interesting question. At the moment, I think the short answer is yes. If you want to do that right now, you would have to kind of manually compare and look at them side by side. But we're looking at different ways in which you could essentially do a diff that way as well. And the kind of converse use case to that is, of course, propagation, because you might want to propagate from a well understood pathway in mouse to what the equivalent pathway might be in human. And of course, we make heavy use of these kinds of model organism to phenotype propagations in Go standard annotations. There was recently a paper, the Nature paper about this, the human functionome paper that showed how how model organism genes our gene function annotations have been crucial to elucidating the functions of the orthologous genes in human, and we definitely want to do similar things for GoCams as well.
The other aspect about GoCams that interests me is there can be cell type specificity in those pathways as well. And is that on the future plan for GoCam models, is to represent differences based on cell type? I'm thinking, for example, of glycolysis in oocytes in mammals. It's different in the oocyte versus the companion cells. So oocytes don't have the hexokinases, so they miss that first step. So they have to import from the companion cells. So there's really an interesting cell type specificity to how those pathways are put together. And that context can be very complicated, I would imagine, to represent.
Absolutely, and that's not something that we're planning in the future. That's something that we're doing right now. So in many of the existing GoCams, you can actually see that the location slot is filled in with not just the Go cell component, but with the actual cell type as well, or tissue type. And I believe we actually do have some of those differential glycolysis pathways as well, probably done by David Hill at JAX. So.
Probably. So one last question. Just curious how gene orthology and parology annotation is connected through the gene ontology. I guess this is our orthologs and paralogs. Do they get the same ontology annotations, I guess is the essence of the question.
Yeah, I mean, yes, the short answer is yes, but we don't do a kind of like a simple propagation across orthologs. We do a very principled phylogenetic propagation, and so I didn't go into too much detail here because this is like more a general ontology talk, but I definitely encourage you to check out some of the Go materials in the recent Functionome paper. that describes our methodology here. But very briefly, the idea is that model organism gene annotations to extant existing model organism genes, a curator will look at those and look at the phylogenetic context, and they will actually annotate to an ancestral gene or ancestral protein, and say, the ancestor of all of these genes in all of vertebrates likely has this function based on what I'm seeing, what the MGI gene says, and what the zebrafish gene says, and what maybe is known in a few other species. And the curator will actually look at this, through a phylogenetic lens, actually looking at the multiple sequence alignment, and say, okay, I believe this function existed in this gene in the base of all vertebrates, and then we can essentially propagate that down in a very principled way to give you highly reliable human annotation, and indeed annotations across all species. So yeah.
Right. Well, we're near the top of the hour, and thank you everybody for attending, and Chris for excellent presentation. And hopefully we'll see everybody in September at Damien Smedley's talk. Thanks all.
Thanks all.